Okay, well, thanks for everyone for joining us. I would like to introduce Dr. Schneider, who will be giving us our CME lecture for today. Dr. Schneider is clinically trained physician scientist who recently joined the cardiovascular medicine department at Mayo Clinic's Jacksonville campus. He obtained his MD and Ph.D. in genetics from Yale School of Medicine and graduate school. He completed his internal medicine residency and cardiovascular diseases fellowship at Brigham and Women's Hospital. He has maintained a continuously NIH-funded research lab and clinical practice in cardiology. After several year-long research sabbatical at Mayo Clinic in Rochester, he recently accepted a position as Senior Associate Consultant in Cardiovascular Medicine at Mayo Clinic, Jacksonville. His research interests include gene editing and cardiomyopathy, and he will be educating us today about COVID-19 and the human heart. Dr. Schneider, we are very excited that you can spare some time of your busy day to give us this lecture, and we are all ears, all yours.
Okay. Thank you very much, Dr. Al-Sheikh. I really appreciate the opportunity to speak to you. I've realized, for the past few years, I was basically doing postdoctoral research in Rochester and didn't really have any clinical work really. I was working on a gene-edited pig. The only clinical work I did really was heart failure in pigs. And I've just realized now, moving to Mayo Clinic in Jacksonville and starting doing ward time and doing clinic, that the COVID-19 story for cardiology, in some ways, even though there may be a sense overall that the pandemic is ending, I think for many of us in cardiology, it's still just beginning, and it's very, very much active and very much an important issue that we see. I've already seen, half a dozen patients coming with these complaints in just this past month, and I think it's an ongoing problem that we all need to have a little bit more knowledge about in terms of dealing with patients. Just to give you where I'm starting with, this is actually the last talk I gave was a year ago, almost exactly a year ago in 2021 for the Mayo Clinic Sports Medicine Summit. And that sort of tells you how COVID-19 has impacted things. But I thought, as I was trying to prepare for this talk for you today, that it's really I could basically use this talk as the starting point. So I've adapted a talk that I originally designed a year ago, and really not that much has changed. What I'm going to do is present to you some of I'm first and foremost a laboratory scientist, so I hope you will indulge me that I'm going to show you some pictures of cells and various things, not too much science, but just some really nice pictures of things to try to put this into perspective and give you the perspective of the research, the ongoing research that we're doing in this area to try to get to some of the really answers to the critical questions. So I have no disclosures, won't be referencing any off-label, although almost everything in this area is at this point still off-label. But what I'm going to tell you, sort of the organization of this talk, is I'm putting it in the context of the youth athletes. For one, mostly because it makes kind of a depressing topic sort of fun to talk about, is how it has an impact. All of us probably have kids or know kids who sort of are involved in youth sports or even semi-professional, professional sports, and this is has become a very interesting area, certainly within cardiology, in terms of allowing participation of sports in kids who have had COVID-19 or have the post-COVID-19 sequelae of myocarditis. So a very important issue in the context of youth athletics. I'm going to start with talking about athletic spread, talk a little bit about cardiac MRI diagnosis of COVID-19, a little bit about the Lake Louise myocarditis criteria, which have had an impact in this area, sports eligibility and disqualification, and then a little bit about what I call the complications for the metaphorical heart, or how this has impacted kids in general who are involved in in athletics, and then talk a little bit about clinical trials at Mayo Clinic. And I'm also going to talk a little bit about vaccine myocarditis, because that's certainly a very important part of this story. And for a lot of it, I'm going to tell you a little bit of the clinical story, and then I'm going to dive a little into the cell biology and molecular biology without getting too, hopefully not boring you with too much stuff, but just some very simple pictures of things. So again, describe COVID-19 spread among athletes, assess the role of cardiac MRI and the Lake Louise criteria, connect myocarditis to disqualification and the ineligibility criteria. And of course, since this talk was designed, it was a year ago, things have really changed, but it's still important for us to understand what the rationale of what the rules were and why they were established, and then reinforce the impact on the athletes themselves. And most crucially, underlying all of this will be to appreciate the biology and the breadth of clinical trials going on at Mayo Clinic and around the world. So as a starting point, let's talk about masks. And please, if anyone wants to interrupt me, I welcome an interruption at any point. Just unmute yourself and let me have it. Starting with masks, I think in the early days, it was shown in a very important study by UW-Madison that masks actually do help in young athletes. This just shows an example of comparing boys baseball to girls basketball. And certainly, although the amount of COVID-19, the incidence of COVID-19 was very low in both of these groups, masks, certainly for girls basketball, made a big difference and basically cut the spread of COVID-19 in girls, which is an indoor sport, right? Cut their transmission in half. This is an image, this is actually my son with his baseball mask at sort of the time when we were, when I designed this talk a year ago, this was him sort of playing baseball, wearing his little, the mask really, for him and many of his colleagues, was more of a decorative thing. It looked really cool, right, rather than actually being a functional mask. But the National Federation of State High School Associations, the Sports Medicine Advisory Committee, came out with a statement on youth sports and COVID-19. And they basically arrived at the conclusion that high school athlete COVID-19 rates tracked precisely with the community rates. So if you were in a place where there were higher community rates, New York or Los Angeles or one of the big cities, the rates among high school athletes was similar to society at large. Non-contact sports were much lower than contact sports. So something like wrestling was higher than, for example, baseball. Outdoor sports were much, much lower than indoor sports, although both of them were still very small. And that was an example I showed of boys baseball versus girls basketball. Indoor, adding face masks to indoor youth sports reduced the rates to outdoor rates, so that you could basically have the example I showed of girls basketball, basically for the girls to wear masks during basketball, that cut their rate, the incidence of COVID-19 in half. And ironically, maybe not so ironically, but sports, almost all of sports related spread in youth sports was due to social contact. It wasn't due to the game itself. So that means locker rooms, sidelines, traveling, dining, interacting. That all of those things is where the transmission of COVID-19 was thought to occur. There was basically, and in particular for baseball, there was no on-field transmission of COVID-19. And I think that they applied generally across most of the sports that they looked at. So it all had to do with the social interactions of the young athletes rather than on the field effects. One particularly amazing place that I know, if any of you are familiar with sort of youth sports, there's this place called IMG Academy here in Florida, where my son had the opportunity to go there a year ago. He actually just went there a few weeks ago again. It's kind of a college for high school sports. a totally amazing place. They have elite athletes from all over the world that come there. And of course, during the COVID-19 time, they had people going home and coming back. And you can imagine it was a nightmare to try to control COVID-19 among their athletes. And they tried very hard to protect and isolate their students, and did a really fantastic job. And Patrick Mulleroni is a Johns Hopkins cardiologist who basically ran their sports COVID-19 program. And it was really, they did a phenomenal job of isolating people who they thought who had positive tests, doing MRIs and echoes and stress tests on people who they thought were at risk of cardiac complications. So, and this all occurred in the context of some studies that were released basically in the beginning of 2021. And the first one you may remember is two of them actually came from Germany. The most famous one was this study called the Puntman study, which was really a shocking result and kind of set not just the athletic world, but the whole world on fire, because what they basically did in terms of MRI diagnosis, they were able to provide evidence that almost half of, they studied 100 patients, mean age of 49, and these patients had a variety of severity of illness, from severe, mild to moderate, to completely asymptomatic, with days to diagnosis after 71, average of 71. They studied them 71 days after the onset of COVID-19, either a positive test or development of symptoms. And independent of how sick they were able to show that 73% had abnormal cardiac MRIs by T1 criteria, 60% by T2, and we'll talk about this in a second, 32% by late gadolinium enhancement. So a range of from 32 to 73% of these patients now, 3 months after having COVID-19, had positive MRIs that would define them as having myocarditis, COVID-19 myocarditis. So that was quite alarming. Another study from Europe also showed similar data, where 54, basically half, and here they studied hospitalized patients who had been troponin positive, and studied them again 2 months after leaving the hospital. And for most of these patients, they were completely fine, right? They thought they were recovered from COVID-19, but when they studied them by MRI, half of them, more than half, still had diagnostic criteria of myocarditis by cardiac MRI. Now, these studies were quickly replicated, and with fortunately more accurate, or more at least we think more realistic data. One of them was done at the Ohio State University, and then there were two, one at UW-Madison and at Vanderbilt. And in all of these cases, they were looking at, because this was a sports, the concern was the athletes. They looked at otherwise healthy young college athletes, and they found much better data. The Ohio State University study was still pretty concerning. They found a lot of patients who had abnormal nuclear studies or abnormal MRIs. But then the two studies that have now been accepted as the dogma were from UW-Madison and Vanderbilt, where they basically showed hopefully much more realistic numbers of 1 to 3% of young kids, right? These are kids that the mean age now was 20, studied out 2 weeks or 3 weeks after COVID-19, that there was still evidence of myocarditis, but it's much less than the original studies that came out of Germany. So the concern was much less, nonetheless important. And I think one of the key takeaways from these studies is this is really the first time that cardiology or radiology has done these type of studies on otherwise healthy athletic kids, right? or young adults. So we really didn't have a baseline of data for what was abnormal, what was normal, and what was abnormal when you do this super high resolution cardiac MRI looking for abnormalities of fibrosis and various things, or edema in the myocardium.
Dr. Schneider, is the
Sure.
timing of the MRI a concern regarding these studies? 1-2 months later versus less than a month?
Yeah, I think that was the major concern initially, is that, for example, in the German study, it was done almost 3 months later, and the patients had gone home from the hospital. They were doing fine, but they brought them back for a follow-up MRI and found that in that case, the majority of them had abnormalities. So certainly that's the concern, is that we're looking at, and even in the US studies, the college studies, it's still concerning that there were abnormalities persisting weeks after the patients were completely fine. And that's what we're going to talk about. What are the long range and long-term implications of COVID-19 myocarditis? So certainly that is a concern. And on all of these studies, there are ongoing longer-term follow-up studies in progress, and we will hear about the evolution of these data. Again, it's important to reiterate that this is a natural history experiment. COVID-19 is a natural history experiment at the very beginning, right? At time zero of the natural experiment. So we really don't know what to expect, but certainly these results, even though they were better, as when repeated, are still concerning. And if you just do the math and think about the numbers in terms of college athletes, if 3% of college athletes had COVID-19 myocarditis by these types of studies demonstrated by MRI, there are 480,000 NCAA athletes in the United States. That would be 14,400 new cases of myocarditis for you and I to follow. So, and we will talk about long-term, right? What does this mean in terms of long-term sequelae? Does this put them at risk for developing dilated cardiomyopathy, and that 10 or 20 years from now, we will see an increased rate of dilated cardiomyopathy that resulted from COVID-19 in 2019, 20, and 21. So I show you this kind of as a joke, really. This may be the only time I've been called a health expert, but I was actually quoted in the Washington Post, and I was interviewed for the story, and I kind of had the story wrong, because I was mostly, at this time, as you could see, my son is a baseball player, and I was very concerned about the spitting that was going on in baseball at every level, right? Spitting is really endemic in baseball. Almost everyone spits, with the possible exception of the umpire, but the umpire even sometimes spit. And you just see it everywhere, right? And this was at a time when we were freaking out about transmission of COVID-19, but it never stopped baseball players, including the professionals, from spitting, and including at the plate. So I was interviewed and had this story, and I didn't get in too much trouble for this from Mayo Clinic, but I don't think they liked my my commenting about this. But this is actually a real issue if you think about it, right? Because even back in the days of the influenza pandemic, the baseball players wore masks, right? We never got to this point with COVID-19, but in the 1920s, right, because they were afraid of transmission of influenza, you can see the batter, the catcher, and even the umpire were wearing masks. Very few of our players at any level in the United States were willing to do that. And then even like we're throwing spitballs at you, right? That could be balls covered with SARS-CoV-2. So the reason this was a grave concern for me, and I went on kind of this rampage, was that you can guess that my son here is the catcher. And this was a video actually from the trip a year ago to IMG Academy. And you can see no one here, I mean, everyone is carefully monitored for signs and symptoms and tested for COVID-19. But you can just imagine the baseball players are always spitting, right? They spit. It's a nervous activity of all baseball players, basically. There are very few that don't, but they can spit on the plate. It's very close to the catcher, which is my son in this case. And more importantly, it's very close to the umpire, which is generally an older man who could very likely have cardiac or pulmonary disease. So it was an issue that never really came to the forefront, but it was something that I was sort of complained a lot about, and it helped me sort of get involved in this area. So going back to MRIs, we won't go into too much detail, but here's just an example of a case of where COVID-19 appears to cause myocarditis with months delay. And I think I've seen already here at Mayo Clinic, being on the wards, even just one rotation on the wards, we've seen patients who come back months later with myocarditis that appears to be related to their COVID-19 infection, or in some cases, COVID-19 vaccine, that we just can't really explain exactly what's going on. This is an example. This was a case given to me by a radiologist in Rochester, a 59-year-old man who initially had COVID-19 pneumonia in March of 2020. He was very, very sick, required ICU care, had a DVT. In April, he had an acute MI. In June, he had continued shortness of breath. Many months later, continued shortness of breath and dyspnea, but his LV function was normal. So it's not as if the MI had caused significant reduction in systolic function. Yet on CMR, he had months later, after this is June and the original infection was in March, there was diffuse abnormalities, not in a coronary distribution, both by T2 weighted imaging and late gadolinium enhancement in his myocardium, which gave him the diagnosis of myocarditis, presumably, although again, unproven, right? And that's one of the challenges of myocarditis. It's very difficult to prove who the culprit organism is, whether it was related to COVID-19 or not. And this is just another example of what you really need to make by what's called the Lake Louise criteria. And the radiologists among you will be very familiar with this. You need both T2 weighted evidence of edema and late gadolinium enhancement evidence of inflammation to really make the diagnosis of myocarditis. But it turns out in many cases, this is such an ultra sensitive test, it's not hard to find evidence of those two things that technically give patients the diagnosis of myocarditis, even though in many cases we don't know what the clinical significance of that is. For athletes, if you give them, if you write myocarditis on the chart, right, that sort of raises this thought or the consideration for sports ineligibility and disqualification. And you can go through, these are the official guidelines. And basically they say that if you define someone as having myocarditis by MRI or even clinical criteria, you need to do echo, Holter monitor, exercise, ECG 3 to 6 months after the initial illness, which for athletes is catastrophic, right? Because that takes them off the field for 3 to 6 months. So this becomes a very important issue for kids in sports and even college and professional athletes, as you've heard. So I just wanted to briefly mention that and I saw this as the father, you know, on a team where we would travel all around the country with this team. This is called the Minnesota Blizzard baseball team. Kind of a funny name, like for a baseball team in Minnesota, but they're very good. And we traveled around the country, and it would have a huge impact on the last few years of kids, including my son, who will be playing college at a D3 baseball. Will be playing baseball at a D3 college in Minnesota. But it's had a huge impact on the last few years of college sports, not only through fear of quarantine if they're test positive, enforced contact tracing, which is a big deal for these kids, mask annoyance, and again, the issue of like someone like me yelling at them to not spit, disrupted time management and training schedules where athletic centers were either closed down or had restricted time scheduling. It, in many cases for the last few years of kids, derailed recruiting efforts and career trajectories, had a huge impact on the last few years of kids, and will continue to have an effect for the next few years. There's been a backflow of college athlete pipelines where kids who didn't get to play because of COVID-19 were given extra years, right? And now all they did was lift weights for the last few years on their own, and now they're even huger kids who are getting into the sports pipeline and sort of taking places away from the younger kids. And of course, kids' skepticism regarding the virus. So now I'm going to switch gears a little bit and talk a little bit about the virus itself and some of the biology of the virus, and how, in particular for cardiology, we need to understand the biology of this virus. It has a particular, unique effect on the heart, even though clinically it may not be as highly recognized. I'm going to present some laboratory data that will hopefully be convincing. So in terms of coronavirus, you know, there are a number of different types of viruses. They've been around for many, many years, over 100 years. I'm going to talk about some of the non-pandemic viruses, which include these viruses that have names for human CoV or human coronavirus. SARS-CoV, which was the first SARS-CoV virus, the first endemic virus in 2003, MERS in 2012, and then SARS-CoV-2, which was unique in that it had a unique furin cleavage motif inserted into the genome of the spike protein. So to give you some of the history of coronavirus, and this is something that truthfully, I didn't know anything about this before I started working in this area. But back in the 60s, when human coronaviruses were first started to be identified, there was a virus called 229E, or HCoV 229E. It was initially studied, and it's horrifying to think of this today, but this was basically the SARS-CoV-2 of the 1960s, in 1967. And they studied it because it was generally a benign, even though some patients got very, very sick with it, it was generally benign, and they would get volunteers where they would actually inject the virus into their nostrils. So they would inoculate volunteer with cultures of virus obtained in vitro. And if you can imagine, would anyone today allow you to inject SARS-CoV-2 into your nose, right, as an experiment? Hard to imagine. But the truth is, when you look at these studies from the 60s, patients got fever. Many of the patients got fever, headache, malaise, chills, rhinorrhea, sore throat, cough. These were the volunteers that had 500 microliters of virus directed right into their nose, actually. And the illness peaked 3 to 4 days after the inoculation. It lasted about 7 days, pretty much sort of a typical SARS-CoV-2 patient now. The other interesting data is that 50%, while 50% of the patients who had this 229E virus injected into their nose developed these cold-like illness, 30% had virus, meaning they could detect the virus in their nose after a few days, but they had absolutely no symptoms. So there was definitely certain people, 30% of people were infected by the virus, but had no symptoms as a consequence of that. And 20% of these patients, now back in the 60s, had nothing. You could not recover the virus from them. And although admittedly, this is a different coronavirus than SARS-CoV-2 or SARS-CoV-1 or MERS, it is very closely related. It's part of the same family. Most importantly and concerningly, there was no stable immunity. They took these same volunteers and a year later injected them with the exact same virus that they got a year ago, and there was no long-term immunity. They could be they could be successfully reinfected a year later. So they did not develop natural immunity to these viruses. So backing up a little bit to myocarditis, it's more common than you'd think unless you're working in this area. There are many viral culprits for myocarditis, and this is the list. You've heard of all of these. The perplexing thing is often when we get tissue samples from either biopsies or post-mortem samples, and you look at the myocardium of someone that you thought had has myocarditis, you can actually isolate multiple of these organisms. It's almost the rule that viral myocarditis is not due to one virus, it's due to many viruses, or at least we can identify genomes, or in some cases, viral particles of many organisms within the same tissue, so that you might find a nidus of lymphocytic infection within the myocardium. And if you look here, you will find, and you isolate the RNA or the proteins or the viruses themselves, you will find there are 3 or 4 different viruses in there. And that's almost universally the rule. So again, there's a lot we don't understand about viral myocarditis, but it's very unusual to be a one organism, one disease activity. It's actually the myocardium is susceptible to many of these viruses that actually live in and around us almost all the time. For some reason, there is a circumstance is established where the virus or viruses can take hold and cause an infection. So it's important, in particular in the context of understanding COVID-19 myocarditis, to really try to understand what we've known about myocarditis for many years. And that the truth is, the true incidence and prevalence of myocarditis is completely unknown. The majority of cases is subclinical, and 60% or 70% of cases is subclinical and undiagnosed, meaning there are a lot of people out there that never come to our medical attention who have viral myocarditis. We never see them. It's a disease predominantly of young to middle-aged individuals, median age about 40, men more than women. By ICD code, it's diagnosed about 1 in 10,000 individuals per year globally. Now, some of the most concerning data that we have is that 1 to 5% of a typical, what we would consider a typical cold that you or I may, would have, especially if it involves Coxsackie B, involves the myocardium. So that if you've had 20 if you've had 20 colds in your life, your chances are you've had myocarditis, and a very good chance you've had myocarditis. You may, and you probably never knew it. And I think this has important consequences for when we think about idiopathic dilated cardiomyopathy, starting with just patients who have acute onset heart failure that is otherwise unexplained, meaning it's not coronary, it's not, they don't have a genetic mutation that we can identify. That's due, and again, these are controversial data because it's very hard to get these data. And some of the studies were done in older times when we didn't have RT-PCR and these various super or genome sequencing high-tech strategies, but 10 to 40% of unexplained heart failure is thought to be myocarditis. 65% of idiopathic dilated cardiomyopathy biopsies contain viral genomes, plural, and 4 to 10% show myocarditis. So myocarditis is probably much more common. There is certainly suspected to be occult myocarditis that travels through human populations that is never brought to medical or cardiology attention. And that certainly will apply to SARS-CoV-2 as well. I'm going to tell you about one particular patient that we saw, and you can look up this paper. It's you can just, unfortunately, you can look up this paper, but this was a patient who was a 35-year-old woman that we didn't actually see her. This happened in Michigan. She presented with sudden cardiac death. This was a young mother from Guatemala who was basically giving her child a bath and told her husband she wanted to go lie down. She was feeling dizzy. She went to lie down on the couch, and 15 minutes later, her husband went out to see her, and she was dead. She had had presumably sudden cardiac arrest, and didn't even know that she had COVID-19. She was diagnosed with COVID-19 post-mortem, didn't really have pneumonia or any other signs or symptoms of COVID-19, but she had clear myocarditis. You can see kind of patchy infiltrates in her myocardium. If you go to this study, you'll see stains for the histological stains. What we were able to do is actually look at her myocardium, and this is actually her cardiomyocytes stained in red, and the green is CoV-2 spike protein. So this is the spike protein from the virus infecting her heart. And you can see that if you look at the myocardium, it's actually, the spike protein is a membrane bound protein, right? Here's the membrane. It sticks outside of the cell, and it goes into the T tubules of the cardiomyocyte, which is the membrane, when you look at the cardiomyocyte, are the membrane structures, right? The outside of the myofibrils. So she had spike protein. She was infected with SARS-CoV-2 and had spike protein in localized areas of her myocardium. So just to show you some pictures of this, based on sort of building on that study, we did a lot of work looking at expression of or infection of human cardiomyocytes with SARS-CoV-2, as well as expression of the proteins, the spike protein alone in human cardiomyocytes. And you can just imagine that this is a human cardiomyocyte. You can tell it's a cardiomyocyte by the striated pattern of its myofibrils. And the green staining here is the Ace2 receptor, which is the receptor that SARS-CoV-2 uses to get in the cells. Here is a cardiomyocyte. Again, this is a cell culture model expressing the spike protein. And you can see these little tentacles, basically, of the cardiomyocyte, that it's reaching out to the neighboring cell, trying to spread the infection through spike protein from one cell to an uninfected neighbor cell. That's how it spreads. I mentioned that it was in the T tubules of the cells, and this woman died of presumably an arrhythmia. Of course, we didn't have any kind of cardiac data on her because she basically presented post-mortem with sudden cardiac death, but these are normal cardiomyocytes in culture. If you express the SARS-CoV-2 spike protein in human cardiomyocytes, this is what you get, and we call this a tsunami spread of electrical, or in this case, it's calcium flux through the cardiomyocytes that in this case are actually fused to one another, creating a multinucleated cardiomyocyte. So this is some of the biology of what the virus or the spike protein itself can do in human cardiomyocytes. And it's important to note that we only see this in human cells, right? The Ace2 receptor, the SARS-CoV-2 virus, only infects human cells. You can't do this study in mice or really any other species, because the SARS-CoV-2 spike protein only binds to the human Ace2 receptor, which limits some of our ability to do this science. But returning to this woman and other patients that we've studied, we have this hypothesis that the virus, cardiomyocytes in myocardium are connected to one another through synthetia. It's technically a synthetia. All cardiomyocytes have intercalated discs where every cell is connected to 3 or 4 or 5 or 6 other cells. So it's basically the heart functions as one big cell, electrically and mechanically coupled, and that the spike protein creates what we call conduits in between cardiomyocytes and allows virus to spread from cell to cell within myocardium. And that's why we saw in the picture of the histology, we saw only clusters, small clusters of cardiomyocytes, as if it was in fact spreading from cell to cell. Just to show you that the human cardiomyocyte is an incredibly good, maybe even the best host for infection by SARS-CoV-2 and production a virus, this is a human cardiomyocyte. You can tell this is the nucleus of the cell, and these are the myofibrils kind of stained in red. These little blue things, and they're not really blue, we pseudo-colored this image. These are viruses. Here's virus on the surface of the cell. They get in, they take over the cell, and basically pirate the cardiomyocyte, which is already a factory, right? Normally, the cardiomyocyte is a factory for making myofibrils. Here, it's been abducted or, pirated by the virus that basically uses the machinery of the cardiomyocyte to make tons of virus. So human cardiomyocytes are very efficient at producing SARS-CoV-2. So that brings us to whether that occurs in vivo or not. We have some, and other people across the world have some data, but the idea is, how do we deal with this? We now have, this is basically a picture of this kind of, all of these things are going on in this cardiomyocyte. This is the cartoon version. And there are a number of drugs, and I'm going to speed up because I want to leave time for questions. Convalescent serum, various Ace2 blockers, losartan, and various chemicals, hydroxychloroquine, certain remdesivir, various drugs that allow us to interfere with the biological life cycle inside and outside of the cardiomyocyte. This is another cardiomyocyte at 50,000 times magnification, right? This is a scanning electron micrograph, and you can see the virus on the surface of the cell. Each of these particles is a virus, the SARS-CoV-2 virus. Again, it's all about the spike protein. And if you look at the surface of these cells, you see these little finger-like projections where the spike likes to go. This is, if you look at this cell on end, you can see them basically a carpeting of the surface of the cell with spike protein that provides the opportunity to design therapies to try to induce immune response against the spike. So one thing right, the Moderna vaccine, I'll go back to that slide in a second. Moderna vaccine is actually mRNA that encodes the spike protein. And we can treat these human cardiomyocytes with the Moderna vaccine, meaning we just take the same vaccine that they injected into you or I, and we put it into the cell culture media of these cells. Cardiomyocytes are actually very good at taking up the vaccine, which is encapsulated in a kind of lipid type vesicle, takes it up and actually makes spike protein. So the intention, of course, is you're injected with the vaccine into your arm. The vaccine goes to the lymph nodes, to the lymphocytes in your arm, and in the arm and sort of under the arm, that provides the microenvironment where an immune response is generated. But there are occasions when, and this has been actually shown in preclinical studies, the vaccine can get into the systemic circulation. If your vaccine gets into the systemic circulation, and again, this would happen very, very rarely, it can be taken up by the cardiomyocytes and expressed as spike protein on the surface of the cardiomyocytes. And here's one particular cell. You can again see it has these little structures, fingers loaded with spike protein. So again, this is in vitro science, very hard, because we can't do this in an animal model. We don't know if this is happening in animals, but certainly raises the possibility. Now, it's important, again, like I mentioned, to sort of understand the pre-pandemic textbook understanding of coronavirus. And we knew, because this has implications for us as cardiologists going forward, even as the SARS-CoV-2 pandemic ends in the next few months, certainly, hopefully, we need to know these types of data, right? That these viruses, all four of these endemic viruses that have infected people back, and this was the one that we talked about in the 1960s, they actually cause up to 30% of upper respiratory tract infections or colds. So 30% of the colds that you or I might get are due to coronavirus, not SARS-CoV-2, hopefully, but other coronaviruses that are circulating in human populations. And all four of these, and many, many variants of them, are still circulating in human population. So coronavirus infection of humans is actually very common. And again, their pathogenic activity peaks every 2 to 4 years. So they kind of come and then go away. They mutate into a less virulent form. Then they mutate into a more virulent form and sort of come back and affect us, usually in winter and early spring. In the context of babies and aged population, especially those with COPD and heart failure or heart disease that require IC level disease, they can have very, very severe, even lethal pneumonia. And again, this is what we've seen with SARS-CoV-2, right? The sickest patients were one that were predisposed to severe illness because of their lung or heart disease. And serologic studies of these other coronaviruses show that people have infections repeatedly throughout life, and that coronavirus neutralizing antibodies are detected in 50% of school-aged children, 80% of adults. I'm not going to talk about SARS-CoV-2 or MERS. These are very interesting stories, but I think it actually is very interesting. And if you're interested in this area, you should go to this recent textbook, right? This was a textbook considered the Bible of virology, Field's Virology. This was written or published in 2021. The word SARS-CoV-2 does not appear in this textbook, but there's lots of information about sort of the what our understanding of coronaviruses, or this entire chapter on coronaviruses, that's really worth reading to sort of clarify some of the misconceptions that we have about coronavirus. And in many ways, SARS-CoV-2, SARS-CoV-1, and MERS were more severe viruses, but the biology is very, very similar. So I'm going to conclude and hopefully leave some time for questions. You know, COVID-19 is an emerging and new pathogen, SARS-CoV-2. Neither acute nor long-term consequences are really understood, in particular myocarditis and what myocarditis might mean for long-term cardiomyopathy. The more recent studies are more optimistic, but it's still very significant for athletes and for all of us, really. Judicious use of CMR is warranted, but it can be very tricky, and it's very easy to overinterpret myocarditis based solely by magnetic resonance imaging. This virus, in all likelihood, is going to affect humanity for a long time, not just SARS-CoV-2, but as it evolves into other related entities. And drug therapies are crucial, as well as vaccination. More studies are needed at molecular, cellular, tissue, organ function, and clinical levels. And I think now, as the pandemic sort of ends, we will have more of an opportunity to study this virus in the laboratory in certain circumstances where we can get answers to some of these questions. So I want to thank some of the people who helped me with this presentation, some of the data, in particular Leslie Cooper, who honestly it was through this work that I was recruited by becoming close friends with Leslie. This is how I got recruited to Jacksonville, to Mayo Clinic at Jacksonville. And we're continuing to work on this, and we'll hopefully have both clinical and basic science studies that will help to clarify many of the outstanding questions in this field. And I'll end there, and thank you very much for your time, and thanks for listening.
Thank you very much.
Okay.
Open for questions, if anyone has any question.
Hopefully that wasn't too much science and too many pictures of cells and things.
No, I think it's great. I have a question, Dr. Schneider. So
sure.
Any data regarding the incidence or the severity of myocarditis in vaccinated patients if they get infected?
Well, there certainly is evidence, you know, again, and it's important to highlight that the benefit of the vaccine outweighs the potential risk. But there, I've been at Mayo Clinic for 3 months. I already have, for 1 month. I already have 3 patients in my clinic who have presumably vaccine-associated myocarditis at some level, at least symptomatically. Again, very difficult to prove causality, but they connect the injection of the vaccine to cardiac signs and symptoms, which can be chest pain. And overall, the data is that yes, less than 1%, but and of course, millions and millions of people now have been vaccinated. So this is again, it's a human experiment, and we are just the natural history has just begun. So this will be something that we will all follow very closely, and has a huge impact on cardiology practice, because these patients are coming to us. I'm sure you've seen these patients as well. We don't understand the mechanism. It could have something to do with vaccine uptake by the myocardium itself. That's again an experimental. The data I presented was laboratory. It's going to be hard to show that in vivo, in a living person, although we're working on strategies to try to image that.
I'm sorry, Dr. Schneider. What I meant by the question, thanks for addressing that question. What I meant is those who are vaccinated and then they get infected, do we have data about the incidence of myocarditis as a result of the disease?
Oh, I see.
Evidence of vaccine.
Yeah, very interesting question. I think it's still, that data, I haven't seen data on that in terms of myocarditis, but certainly we all know friends or colleagues who have had COVID-19 after being vaccinated, triple vaccinated. So it certainly can occur. I don't know if there's any data yet about whether there's an increased or decreased frequency of myocarditis in those patients. That's a very interesting question. That's a great question. I have to add that one to my list of things to study.
Thank you. Yeah. There is a question here in a text format. The question is, will the study data from I think Wisconsin MAD evolve to include longer tracking time, 60 to 70 days versus current 15 to 21 day data, which we discussed earlier?
Yeah, certainly those studies now are about a year old. Absolutely, they're collecting long-term data on all of those patients. And I think the excitement has kind of died down, and we're not getting MRIs on there was a period about a year ago where anyone who came in with this complaint of chest pain automatically got an MRI, and in many cases they were mistakenly read as evidence of active inflammation or scarring, presumably related to COVID-19 or even to the vaccine. So I think it's still, the water is still pretty muddy. And I think it's going to take a few years for us to clarify things. And those studies are certainly, they will get long-term data on all of those patients.
Thank you very much. Any other questions, anyone?
I had a quick question here. So I missed part of the talk. I had to go to another Zoom meeting here, but I'm not sure if you covered this, but one of the considerations in terms of the cardiac MRI data in terms of these athletes is the differentiation between sort of normal pathology versus exercise-induced cardiac remodeling and scar related to that. And then also they've sort of brought up the lack of controls in some of these studies.
Right.
So do you have any comments in terms of
that?
Yeah,
I did mention that and sort of highlighted where the original Puntman paper had a super high incidence of abnormalities by cardiac MRI in post-COVID-19 patients, and talked a little bit about the Lake Louise criteria and how, this is again the first time when we've started looking at cardiac MRIs in 20-year-old kids, right? At super athletic kids. And there was a lot of misinterpretation of things. We really didn't have control data. We don't know what's really normal for an athletic 20-year-old, elite athlete, even high school athletes. So we don't really know what is normal for those kids. We just didn't have the control data. And I think you're absolutely right. A lot of what we were initially calling evidence of inflammation or injury or even troponin leak may just be due to the fact that they are elite athletes and they're working their hearts very, very hard and may have had nothing to do whatsoever with COVID-19 infection or vaccine injection. So those are really key issues. And I think you know, we're reformulating our understanding of how MRI can be used to diagnosis or can't be used to diagnosis. Of course, it's very hard to get tissue, right? No one wants to undergo a endomyocardial biopsy. It's very difficult to image the inflamed myocardium. Troponins can often be elevated in young athletes. So it's a very challenging area, which actually for cardiology, right, it's great because we're going to see a lot of patients and we're going to have a lot of things to sort of work out, study, and sort of figure out a strategy to do this. And again, this is not, even though the pandemic is very likely ending, it's not, the problem isn't going away soon. So, and we may face an even bigger problem with post-viral cardiomyopathy down the road. So yeah, that's a great question. The MRI is still very muddy.
Thank you.
Okay.
Other questions? We still have a few more minutes. Okay, well, Dr. Schneider, that was a great talk. And we really thank you again for your time. We learned a lot and we're looking forward to your next one.
Yeah, okay. The next one will be about a pig, the story of a gene-edited pig with heart failure. It'll have a little bit of this kind of data, but you know, again, lots of pictures and no blots. I promise to not show any blots, just pretty pictures. All right, well, thank you very much for the opportunity. It was great giving a talk again. It's like been a year since I've given one of these talks, so I really appreciate the opportunity.
You've done great.
All right, thank you very much.
Thank you.
Have a nice afternoon. Now I got to go to clinic.
All right, good luck.
Okay, thank you.
Thank you.